This is a real pleasure to be here. I want to thank everyone for the invitation, and in particular, if you haven't had the pleasure to really interact with Joe, he's a gem. So it's a delight to be here. I have a request. Joe can make ground rules, I can't, so I'll make a request. I'm going to stop my talk with exactly sort of 5-10 minutes to go. And in exchange, I really want to get questions. I really do. Because what I'm going to tell you, there's going to be some technical slides, but that's not really what I want to talk to you about. I'm going to walk through and give you my perspectives, and I would really appreciate engaging in a dialogue. So even if you throw out a fake question, there's no such thing as a dumb question in my book. I would love them, and hopefully I'll make the timekeeper happy. There's a gigantic red clock blinking at me back there, so I guess we're on the incentive program here. Anyway, I had the ability to entitle my talk "A Perspective from an Optimist." That is me. And I have to declare that I'm an optimist up front, because just about everything I'm going to tell you is extremely negative. And you're going to say, what's this all about? I thought the guy was an optimist. And there's a reason why I want to do that. And I actually think that these are winnable fights. But you can't win if you don't know the shape of the battlefield. So I'm trying to give everyone here an idea of what that battlefield looks like. And I'll get to what my goals are in a second. Okay. So before I get started, I do want to start by thanking folks. I did mention I was going to stop early on, but as I mentioned, this is an incredible center, and it's a privilege to be associated with it. I have had the good fortune in my life of having some extraordinary mentors. One of them is sitting right there, Herb Conrad, who was president of Roche Pharmaceuticals. And along with him, Roy Vagelos, Ken Ross, a few other folks. It's been a real treat, and it's inappropriate for me not to acknowledge how lucky I've been by meeting some of these folks. We've got a great team at Bridge. We have wonderful inventors. Inventing stuff is hard, and my heart goes out to anyone with the courage to do it. And without question, I really want to thank, I'll do some circling over here, because I'm right in the, it doesn't work, is it? There it is. Our patients. I know we've got some parents and a lot of folks in the audience who know affected people. That's why we're all here. And it's easy to get lost in DNA and assays and things like that. But there's a reason that we do it. And it is a pleasure that I'm able to contribute in this way, because that's really what it's about for me. So let me move forward. Before you understand what I'm going to say, you have to understand a little bit about me. So this slide isn't meant to be self-serving, although it might appear to be. Note what the industry has done to my hair. You get a lot of gray hair in this industry. But I went to school at BC, Rutgers Medical School. I trained in internal medicine at Mass General. I did a fellowship at the NIH. And I've worked through a number of companies that Joe has run through. I've had the privilege of working in a lot of different therapeutic areas throughout my time, and I've worked on a lot of new drugs. But I put this up here for two reasons particularly. One of them is I want to point out nowhere do you see anything about autism up there. So I am the least informed person in this room when it comes to autism and related disorders. So please don't ask me questions about that, because you all know it better than I do, but I do know a little bit about developing drugs. The other reason I put this up here is highly personal. There was a huge turning point in my life right there. So I trained in internal medicine in Boston at Mass General, and it's a 3-year program. And somewhere in the middle of that program, I had a real professional crisis, enlightenment, if you will. I realized it was agonizingly painful for me to treat patients and not see them get better, not see them leave their disease behind and walk away. It was brutal for me to just try to manage patients, and some people are very good at it. I was not. I was terrible at it, and it was frankly heartbreaking for me. And that is what made me realize that my calling was a desire to see if I could be in some way a contributor to making better drugs and maybe changing that trajectory. Even if I could do it once or twice, it was better than nothing, and I could not accept the status quo. So why am I here today? It's a wonderful opportunity. Hopefully I can educate you a little bit. Hopefully I can encourage you, because it's tough work. And my not-so-secret missions here, I'm always out looking for new ideas. I'm always out trying to find what's new and hot and could be translatable. And frankly, this is a movement, so I'm trying to recruit you. Because we need all the help that we can get. And I don't really care where the contribution comes, but this is such a tough game that I think everybody has to be in, and to the extent you can, that's great. Let me see what I can do here. There we go. And this is why. So I've shown this at a number of talks, and this sort of captures it for me. That in essence, we're all somewhat imperfect, and we can only achieve great goals by working together. And that will be a recurring theme throughout. So I'm delighted that Jody Surratz is here, and you'll get to hear from him from Takeda. This really is a team effort that we're talking about here. All right, so what's the big picture? What's that battlefield look like? And what you have to deal with is the fact that there's great research going on all the time, and you're going to hear a lot about that. But when you make a great finding in the laboratory, there's always this question of, is it really a breakthrough, or is it something that's going to fade into oblivion in 2 years and be replaced? There's a translational challenge, and that's sort of where I fit in terms of how do you take these discoveries and begin to build them into a drug? And very importantly, how do you build the collaborations that are going to be required to make that happen? And you'll see why shortly. And this one kills me, innovation, right? I mean, what's the innovation risk? Everyone wants to be an innovator until you get right down to it. These are not made-up statements. These are things I've actually heard. If it's truly novel, then no one has done it before. But if no one's working in that space, then shouldn't we avoid it? Isn't everybody else avoiding it for a good reason? I'm not making this up. How do you become a pioneer if you're looking for someone else to be there in front of you? And the flip side of that, believe me, there's 6 people there. How can we possibly compete? Let's just back out. And the other one is the tools just aren't there. Maybe we're just not ready yet. And finally, the resources, and this is a big theme, and we'll come back to it at the end. Where are we going to get the money? And who's going to take on that risk? And how does it all get paid? Because now I'm going to show you a little bit about what's involved here. So these are statistics that I borrowed from various sources. This one's from Ohio State. And it shows you, if you haven't heard the concept of the gigantic funnel, I think I killed the pointer. Yeah, I did. Okay, so at the top of the funnel is sort of the massive things that might become a drug. And you can see in highlights, it's about 15 years. And even more daunting, about $2 billion is the current estimate. I can remember when I started in the industry, and that estimate was 500 million. It's now gone up to $2 billion. Okay, so here's a different way of looking at that same thing. We have a lot of things starting at the front, and what you can see is very few things come out the back end, and early on there's a lot of risk, but that risk never goes away. And how would you feel if you were the person that forked over $1.5 billion only to find out it didn't work? So, this isn't intended to bash anybody. It's intended to understand why it's so tough. There's a reason why it's tough to get partnerships done. There's a reason why it's tough to get traction. This is an enormous risk, and frankly, you question whether or not sane people would actually do it, knowing that. But it's a big challenge in order to get something to work at the back end. And here we have the cost. In terms of the cost goes up, it starts small and then builds exponentially as you go forward. So while that cost is building exponentially, you're still not out of the woods yet. You may have some great data from the laboratories, but failure is lurking around every corner. And that, I think, is part of the problem. I'm going to skip through this. We've arbitrarily drawn lines about these different phases, and if anybody's interested in the technology and the jargon. I'm happy to talk about that. But I did want to make the point that the understanding of the biology never ends. So the research work is really important throughout the process. All right. So I'm going to focus a little bit in this talk on the very early steps, the quote-unquote finding the drug step and finding what that asset might be. And just to highlight, the amount of time involved here, we're talking about, 2 to 5 years to get to where you need to go. Joe mentioned I worked up the street on the Upper East Side, and this is our model. We basically work with 3 institutions, and we look for opportunities to translate those ideas into drugs. So it's a partnership experiment that we're working with, and I may talk a little bit about that as we go forward. I'm glad I thanked Jordy. This is his slide, so I hope Jordy isn't using it. But Jordy, when you lend me a slide, this is the risk you take. So anyway, this is from Takeda, and it describes a little bit about how they go after making a drug. And it's not unique to Takeda. There are some nuances with every company. But you start by identifying your target. If someone has yet another pointer, it would be great. You start by identifying your target. What is that gene target that you want to hit, for example, or what is that cellular receptor that you want to hit? And then you have to develop assays by which you can screen to see if your new drug is actually hitting that target. Look at that. Magically, it came back. HTS stands for High-Throughput Screening. That just means you got a barrel of potential drugs. And some of these barrels contain 500,000, 1 million, 2 million compounds. And think about the amount of time involved in order to screen those. And that's why that issue of high throughput is so important. Because if you were doing this with a technician one at a time, you'd be at it for several thousand years before you had your first set of results. And no one can do that. And in the end, I killed it again. In the end, you get something called a tool compound, which means it's okay for exploring the biology, but for one of 20 different reasons, it's never going to be a drug. But you can use that tool compound to do something really important, and that is this concept of target engagement. I mentioned the target up front. Now you've got a molecule. How do you know that that molecule is actually affecting that target? And that's why people in the industry use this term, target engagement. Are you actually doing what you think you're going to do? And if all that works, you go through a screening cycle that leads you to potentially a drug candidate at the end of the day. So I'm going to get at a concept that if you've ever been in a meeting, and Joe, I love that analogy of the made-up syndrome. That was great. But you get into these questions of drugability and how do you manipulate a target. People use this term drugability. It's really, really important, and it sort of combines this concept of the science is cool, your molecules may work, but is it a druggable situation? Can we potentially make a drug at the end of the day? And it's a guess, and think about that. Think about walking into a casino with limited information and plunking down $1 billion bet on the table. It's hard to do, which is why there's a lot of thinking that goes into it. And Joe touched on a lot of these aspects in terms of what is the disease? Can I define the disease? Because sometimes you just have a constellation of symptoms, but everyone asks, is it a specific disease? Are there 40 diseases that all look the same? And then what's the biology? This whole thing about, do you know the cause? Or are you just looking at something that tends to be associated with it? And if you hit that target, given all the risks, is that ultimately going to change the patient and make that patient better? Because that's ultimately what you're trying to do. From a drug perspective, big words, pharmacokinetics. I happen to like pharmacokinetics. We'll get to it later. I'm an affirmed geek, so this is revenge of the nerds when you go into drug development. And kinetics basically means I can measure the concentration, the level of that drug at the site of action. And can you get the drug to where it needs to be? And then the pharmacology is the next question. Well, once it's there, is it doing what you hoped it would do? Are you actually changing the biology? And a really important question, is it practical? Because you can come up with things that can work in a lab, but you can never do with a patient. So is it something that at the end of the day is going to be practical and manageable? Because a lot of people have to deal with that bottom bullet, as painful as it is, the logistics. What's this look like? How long is it going to take? Are we going to be able to make it through at the end of the day? And no one's looking for a 100% assurance. And we're going to come back to this at the end. I don't believe in conspiracy theories. There's no malintent here. But what you do have is a huge spectrum in terms of risk tolerance. And you'll find out very early on that certain people are willing to take on a bit more risk than others. And everybody sets that fulcrum in a different place. I've been told I'm a pretty aggressive guy. I am. I will take on more risk, but not everything. So I set my needle in a certain place and others set them in a different place. And it's not through malintent, especially when the stakes are this high. So I want to touch a little bit on the partnering. And without the jargon, I want to emphasize that there's a real opportunity for people to share their skills in terms of what they're best at. And academic institutions working side by side with folks with industry experience is actually a good cocktail. And I know that point may be controversial, but what we're really looking to do is get people with different perspectives at solving a very complicated problem. And the thinking that goes on, there is no substitute for the kind of work that Joe has shown you. It is state-of-the-art work. And it goes on throughout institutions, great at understanding disease mechanisms in terms of finding that target, in terms of understanding what's the variability around that target. And what does industry do? Industry can think in terms of, well, what are some good models to test it in? Are there similarities? Are there paths that may give us learning? Do we have experience from something else that may be relevant to the specific problem that we're trying to solve? As well as those logistic considerations that go under fancy words like tractability. And I wanted to touch a little bit on genetics because genetics is hugely important. And this was a provocative slide made by a very, very good friend of mine. And his argument was that, genetics really does help the effort. It's not perfect, though. So this is presenting both sides of the coin here. And if you look at certain drugs, as you go up, that means that there's genetic models that validate that you've got a target. And if you go down, that means we had no idea from the genetics, and the drugs were developed without any genetic insight. And there have been successes on both sides of that in terms of very successful drugs like Proscar and Preluent, that had great genetic validation, but it's not a guarantee. So wouldn't it be wonderful if we had a guarantee? You never get a guarantee, and I guess that's the point here. The other point on this slide, though, is that if you don't have genetic validation, the risk of being in that bottom right corner of the numerous drugs that are too numerous to list, that had no genetic validation and ultimately failed, is enormous, and that's what you're trying to avoid. So it really does help. As imperfect as these tools are, they really do help. And I would argue, in the modern era, this is the best starting point of all. So I commend Joe and his team for what they're doing. So I went to the definitive source of the drug discovery cycle. It's called Wikipedia. So this is straight off Wikipedia. And if you look up the drug discovery cycle, this is what you'll find. And I'm not going to walk you through this. Let me see if I get lucky and the pointer works. No. Okay. But there's a path there if you follow. If you go from compound collection, remember I told those big barrels with like 1 million molecules in it? If you go from that compound collection into the primary assay, and then you make that right turn over to secondary assay, there's an arrow that drops down the clinical candidate. Okay. Joe just told me he does have a drug that came down that category, so kudos. So that path that goes down the outside right-hand side, that never happens. Maybe it happened once. I've learned that you can never say never, right? But, fishing out of the barrel and finding a drug just doesn't happen, and it certainly doesn't happen in the modern era. The other thing that's hidden in here, if you go down the more likely path, notice all the circular loops in there. So there's a huge number of potentially circular loops. That does happen, and that happens, unfortunately, all the time. So you go down, you go to an assay, you run into a problem, you circle back, and you keep doing that. And sometimes you do that for 10 years until people get fed up and say, this is never going anywhere. So unfortunately, that drug discovery cycle, it's more dominated by the circular loops than it is by that quick shot to the clinical candidate. All right, talking a little bit about what the assays look like. What we want to achieve is the ability to do this at quote-unquote industrial scale. So the more muscle that you can bring to that equation, the better. And the tools have gotten really good, a lot better. So let me go forward a little bit in terms of what we've got now in terms of the different platforms. So there's a lot that can be done in terms of the screening assays. And in terms of once you find your target, how do you hit it? There's been a real revolution. This is only scratching the surface. We have traditional drugs that come in pills. We have biologics, these infusion therapies of proteins and antibodies. Now we have cellular therapeutics. If you've ever heard about CAR T for cancer, we can engineer the immune cells to attack things. And there's some amazing things, too, on the engineering side with pure electronics that makes this work. So there's a lot of capability to do things, but I do emphasize what's in the title, and it all comes with a rather hefty price tag. So I'm going to show you one or two things that I have the privilege of having access to. One of them is these computational drug discovery suites. It's really cool. I mean, that funky thing on the right, there's probably a few people who know what that is, but that's sort of a model of a protein. So if you identify your quote-unquote target and it's a protein, and then you can make it crystallize, so it precipitates out and you got a rock full of protein. You can send that to one of these laboratories that can do what they call structural biology, and they can determine exactly what that looks like in three-dimensional space, and then they can find the pocket that you want to hit. So, this is a Star Wars kind of stuff. This is when it works. It's awesome. It's really cool. And then, you can use a computer to design what the drug should look like that's going to fit exactly in that pocket. If only life were that simple and it worked every time, but this is really cool and it does happen and it does work. And I think most would say it's an important starting point, but it hasn't solved all the problems. Similarly, there are now a host of engineered animal frameworks. This is a mouse that has been taught to make human antibodies. So I know of a mouse that makes human antibodies. Actually, there's like 3 or 4 of them. There's a cow that makes human antibodies. There's a llama that makes human antibodies. And why do that? Well, because then you could vaccinate that animal and you can get actually a human antibody out of that animal, and it really short steps the path. So there's some mind-blowing good stuff in here, too. See, I'm not always a pessimist. I'm going to skip that in the sake of time. I would say this is about actually physically manufacturing stuff and actually making chemicals or making an antibody. And one note of encouragement here, just in my experience, when you get to an engineering problem, I think humans by nature, or maybe our understanding of science, we're way better at engineering in terms of being able to solve engineering problems as opposed to biology, which is really complicated, and there's so much that we don't know. But I had a good friend in one of my first jobs, and he said, he goes, if you give me a problem in terms of how do I make a bucket of, the drug, he said, I can guarantee you that we can solve that problem. I know what the outcome is going to be. I just can't tell you exactly when it's going to occur. And that ain't so bad. So maybe it takes you a little longer than you think. But it's good to know that from an engineering perspective, we really are good. I don't mean me, I mean human nature. And a little bit about the FDA. I've given this speech a number of times. The FDA is actually a collaborative group. I personally have never gone to the FDA and run into a massive roadblock. They look at things from a different perspective. They are primarily charged with ensuring patient safety. But believe me, they are heroes also, and many of them want to see better drugs made. I've hit my 5-minute mark. Should I?
Yeah, I would a lot more time.
Okay. I'm going to take then 5 bonus minutes. But be thinking of your questions. I'm coming down the home stretch here. Okay. All right. So I do find them very, very helpful. But they're extraordinarily busy. So they don't have the time to meet with everybody who has a good idea. And you have to be very, very thoughtful about how and when to use them. I'll spend a little bit talking about what we're doing on the Upper East Side. So our mission at Bridge is to work with new emerging science that is clinically important in an effort to try to make a new drug product. And we also view a big part of our challenge to be growing a very vibrant ecosystem here in Manhattan, because there's great people and we're trying to foster that environment. So here's a little bit about how we work, and I'm not saying it's perfect, but it's one idea. And what we're trying to do is we're trying to work on that stage where somebody gives us a molecule that they think is good, and we start doing all the early FDA-type work in order to enable clinical trials to get done, including filing the paperwork. I promised I wouldn't use jargon, and I did. So IND stands for Investigational New Drug. That's your application to the FDA, and that application, if approved, enables you to do clinical studies on patients. And then we move it to early clinical studies. So what we're looking for are inventors, and TDI is our sister organization. It stands for the Therapeutics Discovery Institute. So it's a group of chemists and biologists who actually make that molecule. So we've got a target, we've got a molecule, and it leaves from the Edisonian light bulb going off of Eureka. We think we've got something, and we bring it into Bridge. We work on it inside Bridge, and then we try to get it to a point where it can stand on its own feet, and we can ultimately spin it out as a new company. And the reason that there's a need for the Bridge-type entities of the world is because of the money factor, and nobody wants to spend the money on the phase of development. So we take all the unloved children, right? And we're working to try to get them from a point where it's too early for other partners to want to take it on, and then advance it to a point where it's gone to finishing school and it's groomed, and now it's ready to go out on its own. So that's pretty cool to do once, but it's really cool if we succeed and we can do it over and over and over again. So that's a little bit about what we're doing. So I am going to bring it home here with closing thoughts. So do think of your questions, and maybe I'll stimulate 1 or 2 more. So this is what it feels like, okay? This is the ultimate David versus Goliath kind of battle. You are up against Goliath, and it begs the question of why fight, and we'll get into that in a second. But it really, really does feel like a sane person would just give up and not take on the battle. And I'm here to encourage you not to let that go. And here's why. Because I've seen it with my own eyes. And this is just one example that occurred in my lifetime, okay? So I was in medical school in the middle of the AIDS crisis. And if you were old enough to be there at that same time, there was a death sentence. And we lost Freddie Mercury, the lead singer of Queen, as a result of HIV and AIDS. And then Magic Johnson announced that he had the disease, shortly thereafter. And we all assumed the same. But guess what? The whole game changed. And the whole game changed because of what people were able to do through research, through science, through drug development. And now it's entirely different. So these are the things that keep me going, and these are the things that give me hope that it's okay not to accept the status quo. Even though it feels like it's an endless struggle, I can't predict where the outcomes are going to be. That's the point. You can't really predict where the outcomes are going to be. We're smart, but sometimes you need to get lucky too. And yet miracles have occurred. And more recently, you could look at Keytruda and Opdivo and CAR T therapy. There are people alive today who would not be alive, and a lot of them. And that's through the efforts of dedicated folks like you who just wouldn't give up. I'm there with you. And here's another reason to be hopeful. So the tools that we use are getting progressively better. So I talked a lot about high-throughput screening. This is an article that came out within the last 3 weeks, and it's talking about the use of artificial intelligence, which, okay, gets a lot of hype and everything, but this also is a reason to be hopeful. So using artificial intelligence, a group of scientists were able to design a drug in 46 days, okay? So it doesn't mean they can do it every time, but the fact that they could do it once in that kind of time frame should open your eyes. And I'll be the first to acknowledge that, media makes all of its sales by, shocking, provocative headlines. So, discount it as you will. But this is real. It doesn't mean it's perfect. It's in its early stages. Maybe it will fail. Maybe it won't. I hope it doesn't. But that's unheard of. It just doesn't happen like that anymore. So it's good to see that there are folks doing this. And I'm going to close with a few final quotes. This is something I came across. I do not know Jackie Novogratz, but she's starting to say things that really resonate with me. And it's sort of building on me, so she's the kind of person that I want to understand a little bit better. This is a comment that really hit me when she said, cynicism should not be confused with realism nor wisdom. It gives permission to our basest instincts of fear, pulling inward, hardening the soul. It is simply an excuse for inaction. And that's the world that I live in every day. So you can talk about risk intolerance, but I question at times whether or not we've slipped into this mode of it's easier to be cynical. It's easier to play the favorite and bet that nothing's ever going to change. I just showed you that it can. So there's something wrong with adopting a mentality that says, why bother? Everything's going to fail. We shouldn't even try. It doesn't mean that everything's going to work, but I love that she's unmasking what's really going on here, and that is because of my cynical attitude and because I have an IQ over 200, I don't, but I sit in rooms with people with IQs over 200, and they come up with great reasons why we shouldn't do something, and it's frustrating because on a certain level, and again, there's no malintent, I wonder whether it's a question of risk tolerance or cynicism. Because I would argue that you have to stop and ask yourself those questions at certain points. So Jackie's got my vote for this one. And finally, I'll close by pointing out that it's teamwork and it's a race. So a lot of people need to get involved in this. And I'm going to close with, I told you I was going to lay out what the battlefield is. I think you face two major enemies here, and one of them's obvious, and it's money. Okay? It takes a lot of money to move stuff forward. It takes a lot of money to find the target, and it takes a lot of money to generate that. So your big enemies are money, and the hidden enemy is cynicism. So there are other things that we can talk about, but those are just some closing thoughts. I welcome questions, so I hope I get some. Thank you.